So hello to all of you. I'm going to give you the instructions for the Enzyme Kinetics lab. And before I give you the instructions for the lab itself, I'm going to give you some background about enzyme kinetics. So in the lecture, we have already gone over what the enzymes are, how they work, what are the different factors which affect the enzyme activity. And we have also discussed about the effect of substrate concentration on the enzyme activity. So basically, as we have already discussed that when we increase the substrate concentration, initially there is an increase in the enzyme activity, but if we keep increasing the substrate concentration more, the rate of reaction does not increase more, and it reaches a state where it becomes constant, and the curve is like a hyperbola. So we have discussed all these things. So today we are going to discuss more mathematically how we can actually calculate all these things and what are the parameters which can tell us about the rate of reaction. So we are going to discuss about all those things, and then I'm going to go over the lab exactly how you're going to do. So we know that like initially when we increase the substrate concentration, the rate of reaction increases. But like if we keep on increasing, then the enzyme actually becomes saturated and the rate of reaction does not increase more. And at that point, when all the active sites of the enzyme are saturated or they are blocked by the substrate, at that point, the only way to increase the rate of reaction is by increasing the concentration of the enzyme. So you can add more enzyme, only that's a way to increase the rate of reaction. So I'm going to explain you how it all works with the help of an analogy in which we are going to consider a situation where we have some monkeys. So monkeys are representing enzymes and the peanuts are going to represent our substrate. So we have a room of monkeys, which are our enzymes, and they are going to find the peanuts and they're going to shell them. So the peanuts are like the substrate. So this analogy is going to make you understand how an actual enzyme catalyzed reaction takes place. So suppose there are 10 monkeys equally adept at finding and shelling the peanuts. And also imagine a room with peanuts equally distributed on the floor. In each case, the monkeys start out in a room beside this peanut gallery. To start an essay, the door is open, allowing the monkeys to enter. So now let's consider the initial concentration of the peanuts is 1 peanut per square meter. So there are 1 peanut per square meter. So every monkey averagely spends around 9 seconds to find the peanut. And then it takes 1 second to shell it. So which means that in total is spending seconds to find and then shell it. So the shell of 0.1 per second. Okay. But because there are 10 monkeys in the room, so the velocity of shelling is 1 peanut per second. I hope you're understanding how I'm saying all those things. So this is the our first essay in which the concentration of peanuts is 1 peanut per square meter. And then the time to find the peanut is 9 seconds and the time to shell the peanut is 1 second and total is 10. So the rate of shelling per monkey per second is 0.1, but because there are 10 monkeys, so the velocity is 1. So suppose now we increase the concentration of peanuts. So basically keep thinking about this is an enzyme catalyzed reaction. The monkeys are enzymes and the peanuts are the substrate. So now there are 3 peanuts per square meter. So we are increasing the concentration of the substrate. So now what's going to happen? Because now there are 3 more times peanuts in the room, the monkeys are going to spend 3 times less in finding the peanut. So which means that averagely every monkey is going to spend around 3 seconds to find the peanut, and it is going to spend 1 second in shelling it. So in total 4 seconds every monkey is going to use to find the peanut and shell it. So the velocity of shelling is now 0.25 peanut per second for each monkey, and because there are 10 monkeys, it's going to be 2 peanuts. It will be around. So see here, as we increase the number of peanuts, the rate of reaction is increasing. So earlier it was 1, now it is 2.5. So similarly, in the enzyme catalyzed reaction, initially when we increase the substrate concentration, at that time the rate of reaction increases. Now, as you increase the peanut concentration, the time to find peanuts decreases, but with diminishing results. With the diminishing returns. The time to shell the peanut never changes regardless of the concentration. So basically, which means that you may increase more peanuts in the room, which means you may increase more substrate in in the enzyme catalyzed reaction, a point will come when the rate of reaction will not increase. So in the case of monkeys, you may increase more peanuts, but there is a minimum time which will be used by every monkey, which is 1 second, which every monkey will use to shell the peanut. So you cannot decrease that. So which means that even if you keep the peanut in front of the monkey, but still every monkey is going to still spend 1 second. Every monkey is going to spend 1 second to shell it. So which means that the rate of reaction cannot increase beyond 10 peanuts per second. And this is the same situation in the enzyme catalyzed reaction, that when we increase the substrate concentration initially, the rate of reaction increases. It keeps on increasing, but at some point it reaches to the maximum. After that, it is going to stay constant. And at that point, if you want to increase the rate of reaction, you can only increase by adding more enzyme. So this is actually, this thing was studied by two scientists, Michelson and Minton. So on the basis of that, it is called Michelson and Minton kinetics. So most enzymes discuss kinetics. Initially, that's brought by on the substrate concentration. Low substrate S will double the rate of reaction, but as substrate increases, each additional increase in substrate results in a smaller increase in the rate of reaction. When substrate concentration becomes very large, the value of rate of reaction reaches a maximum. So this is the curve to show Michelson-Milton kinetics. So like on the x-axis is the substrate concentration, and on the y-axis is the initial velocity or rate of reaction. So you can see that when we increase the substrate concentration initially, the rate of reaction increases. It will keep on increasing, and then at some point it will become constant like this. Okay, so it shows a graph like this. So at this point, when the rate of reaction reaches to this point, this rate of reaction is called Vmax, which means this is the maximum rate of reaction. Okay, so we are going to talk about what is Km, what is half Vmax, what is Vmax. Vmax is the maximum rate of reaction when we keep on increasing the substrate concentration, and this is the equation by which we can calculate the rate of reaction at any point. So we need to know what is Vmax, what is Km. Using the formula, we can calculate the reaction at any point. So as the substrate concentration, V approaches an upper limiting value, the maximum velocity. So as you keep on increasing the substrate concentration, the rate of reaction reaches to Vmax. The value, so at this point, if you want to increase the rate of reaction, you need to add more enzyme. So the value of Vmax can be increased by adding more enzyme. So this is basically just showing you the enzyme catalyzed reaction in which substrates get converted into products. So first of all, enzyme goes and makes the complex with the substrate, and it's called ES complex, and then that changes to the enzyme and products. You already know that. So this is the way how the reaction goes. E plus S forms ES, and then E forms, then products are formed, and they leave the enzyme. And these are basically the rates of reactions. So the above model under steady conditions gives a Michelson-Milton equation, and according to the Michelson-Milton equation, we can calculate the rate of reaction at any point in the enzyme catalyzed reaction by making use of Vmax and Km. So the most important is the Km, because that's going to reaction. So this is what Km is that concentration of substrate that gives half maximum velocity. So you can see here, Km is that. So this is Vmax, so maximum rate of reaction. Km is that substrate concentration at which the reaction achieves half Vmax, which means Km is that value of substrate at which the rate of reaction is half Vmax. So we are now going to understand the relationship between the rate of reaction and substrate by considering three cases. So you need to keep in mind this equation, that V is equal to Vmax multiplied by substrate concentration divided by Km plus S. And we are talking in terms of this. Okay, so now I'm going to talk about three different situations: when the substrate concentration is very less, when the substrate concentration is very high, what happens at how reaction rate. So in this case, substrate ratio is very, substrate concentration is very less, which means that Km plus S is equal to Km, because this parameter is very small. Because this parameter is very small, Km plus S is equal to Km. So in this equation, in this formula, you can see V is equal to V max multiplied by S divided by Km plus S. So when S is very, very small, when S is very, very small, then this value is actually Km, because this is very small. So the relationship becomes V is equal to V max multiplied by S and divided by Km. So what does that mean? What that means is the rate of reaction is actually proportional to the substrate concentration. So at a very, so the mathematically this formula becomes V is equal to V max multiplied by S divided by Km. So you have to understand the relationship of rate of reaction and substrate concentration. So at a very low S, so when the substrate concentration is very less, at that point the initial velocity of the reaction is proportional to the substrate concentration. So at that point you add more substrate, the rate of reaction is going to increase. That's what it means. But this is a mathematically, this is in the terms of mathematical equation. Now when the substrate concentration is very high, okay, and keep thinking about this this graph. So when the substrate concentration is very less, as you increase the substrate concentration more, the rate of reaction increases. Now we are talking about if the substrate concentration is here, is very high, what happens at that point? So when the substrate concentration is very high, it's very, very high than Km, then the addition of Km plus S is actually S, because now Km is very small. So in that case, mathematically it will become V is equal to V max multiplied by S, and this is S. So you cross this, so V is equal to V max, so which means that at a very high concentration of substrate, the rate of reaction is V max, which means the rate of reaction has attained its maximum value. So which means if the substrate concentration is at this point, the rate of reaction is V max, it's here. Okay, that's what it means. And last one is, and often what the case when the is at that point, the rate of reaction can only be increased by adding more enzyme. Now when the substrate concentration is equal to Km, okay, when substrate concentration is equal to Km, so when the so again going back with this, when the substrate concentration is equal to Km, at that point Km is also S, then it becomes 2S. So you cross this, so V is equal to V max by half. So which means that Km is that concentration of substrate at which the rate of reaction is half V max. So this shows that Km is the specific substrate concentration at which the reaction proceeds at 1/2 of its maximum velocity. So now why Km and Vmax are very important? So Km value tells us the concentration of the substrate at which the rate of reaction becomes half the Vmax. So lower the Km value for a given enzyme. So the lower the Km value for a given enzyme and substrate means that the lower the substrate range in which the enzyme is effective. So lower the Km, which means that at the lower concentration of the substrate, the enzyme is going to be more effective. And Vmax is the maximum rate of reaction. So these two parameters are very important, and Vmax can be used to determine turnover number also. And turnover number is the rate at which the substrate molecules are converted to proper enzyme. Some enzymes are okay. Now, basically, as we are discussing that Km and Vmax, they are very important parameters of enzyme catalyzed reaction, and we need to know them. We need to calculate them. By Michelson-Milton equation, we cannot calculate them to get the numerical values. So to calculate the numerical values, we use another equation. It's called Line-Weaver-Burke equation. And this Line-Weaver-Burke equation is basically an inverse of the Michelson-Milton equation. So it is 1 by V and Km plus S divided by Vmax into S. So it is the inverse of the, so basically our Michelson-Milton equation was V is equal to Vmax multiplied by S divided by Km plus S. So it is inverse of that, and you can see how mathematically it is broken down. So you can break these parts like Km divided by Vmax into S plus S divided by Vmax plus, S divided by Vmax into S. So it can be calculated like, so Km divided by Vmax multiplied by the inverse of the substrate concentration, plus this can be cancelled, plus 1 by Vmax. So this is the line we you can see if we have the y-axis, we have 1 divided by V. So basically it is inverse of this graph, which is your Michelson-Milton equation. In this, the substrate concentration is on the x-axis and rate of reaction is on the y-axis. In this, it is the inverse of that. So basically, it is 1 divided by S, inverse of substrate concentration on the x-axis, and the inverse of rate of reaction on the y-axis. So this is the kind of graph we get. This is the slope. Slope is Km divided by Vmax. And now exactly what we are trying to understand from this graph is that with the help of this graph, we can calculate x-intercept and y-intercept. What is a y-intercept? Y-intercept is that point on the y-axis where this trend line is going to cross the y-axis. And x-intercept is that point. X-intercept is that point on the x-axis where this trend line is going to cross the x-axis. So we need to know this value and we need to know this value, because if we know this value, x-intercept, this is a mathematical thing which you need to remember. X-intercept is minus 1 divided by Km. So which means that if you know this value, at what point this trend line is crossing your x-axis, that is the value x-intercept you calculate. If you know where this y at, and that is to take. So with the help of that, we can calculate the Km and the Vmax. And now I'm going to quickly talk to you about the experiment we are doing. And in this experiment, how to use the graphs and all that, everything is there on D12, but I will be telling you more about those things in the lab. So basically, let's go over. So this is all theory which I've just explained, written here. You can go over it quickly. Yeah. In today's lab, we will look at the effect of pH on the activity of enzyme called mannitol dehydrogenase. So we are actually going to study two things. In this lab, we are going to study about the effect of substrate concentration as well as the effect of pH on the activity of the enzyme called mannitol dehydrogenase. So that's our enzyme. And the substrates are mannitol and NAD+. These are our substrates. And the products will be NADH and mannose. And what is the rate of reaction? You are going to calculate by measuring the formation of product. So the products are NADH and mannose, and we doing the which FC3. So basically this you are going to the concentration and the effect of pH on the activity of enzyme called mannitol dehydrogenase. The substrates for this enzyme are mannitol and NADH, NAD+, and the products will be NADH and mannose, and you are going to study the rate of reaction by measuring the formation of the product NADH, and NADH absorbs light at 340 nanometers. So which means that for this experiment, you are going to use the wavelength. You're going to set up the wavelength 340 at your spectrophotometer. Okay. Now I'm going to explain you the setup of this experiment, and all the calculations and all that I will explain you in the lab. So these are some of the precautions you have to take, and also you will be calibrating the spec as I have taught you in the spectronic lab. So you will be taking, so this is the setup the lab in which there will be basically you are going to set up for 8 cuvettes, and there will be one reference cuvette too. So basically you will be having 9 cuvettes, and let me explain you what these cuvettes are. So you have, this is your blank. With the help of this, you are going to calibrate your spectronic, and these first 4 cuvettes are for pH 9.5, and then these 4 cuvettes are for pH 8.2. So what are you going to add in all these cuvettes? It's written here, but let me explain you. So in your reference cuvette, you will have 2.4 ml of pH 8.2 buffer. So you will be given all these things. So the buffers will be given. The buffers will be at pH 8.2, 9.5. So in the reference, you're going to add the buffer 2.4 ml of pH 8.2 buffer. and you are going to add 500 microliters of 2 millimolar mannitol, which is your substrate, and 500 microliters of, sorry, 50 microliters of NAD plus. And then you are going to add, in the reference, we are not any time, in the 50 microliters of that, you have to add that. The reference, you are going off the spec. And then you have these 4 cuvettes, which are at pH 9.5, and then these are the 4 cuvettes which are at pH 8.2. And you can see here, all of these cuvettes are going to get some of the things which will be common. So like this is for pH 9.5. So all these cuvettes, all these 4 cuvettes are going to get 2.4 ml of pH 9.5 buffer. So the buffer will be same in these 4 cuvettes. Then you are going to add 500 microliter of 2 millimolar mannitol here. Okay, 50 microliters of NAD and 50 microliter of enzyme. Okay, now in the second cuvette, you will put the same buffer, because this is again 9.5. So buffer will be 9.5, 2.4 same. So in all these 4, they will all get the same buffer. They will all get 50 microliter of NAD. They will all get 50 microliter of enzyme. But what is the difference between these 4 cuvettes is, the first one is getting 50 microliters of 2 millimolar mannitol, and this is getting 500 microliter of 5 millimolar mannitol, and third one is getting 500 microliters of 20 millimolar mannitol, and the last one is getting 500 microliters of 50 millimolar mannitol. So what we are changing in these 4 cuvettes is, we are first getting second one and the 40. And this is at pH 8.2. So which means that in this you will be adding 2.4 ml of buffer, which is at pH 8.2. And other than that, so these 4 tubes get the buffer pH 8.2, and they will be getting 50 microliters of enzyme, 50 microliter of NAD, and the first one will be getting 500 microliter of 2 millimolar mannitol. All these 4 tubes will get everything same. The only difference will be in terms of the substrate. The first tube will get 500 microliter of 2 millimolar mannitol. The second will get 500 microliter of 5 millimolar mannitol, and the third one will get 500 microliter of 20 millimolar mannitol, and the last one will get 500 microliter of 50 millimolar mannitol. So this is the way you're going to set up. Very important thing to know here is, once you add the enzyme, the reaction starts. So what you have to do in all these cuvettes is, so add the enzyme at the end, because you have now, this is your reference by which you're going to calibrate the spec. You have 8 cuvettes, and you have to study the enzyme kinetics in each cuvette for different time periods. So if you go beyond here, come down here, you see there's a table here, and given you each for this much time periods, starting from 0, 5, 10, 15, 20. So basically, once you add the enzyme in, suppose this is your pH 8.2, and this is a cuvette with 2 millimolar substrate. This is a cuvette with 5 millimolar substrate, 20 and 50. pH 8.2, pH 9.5. So once you add the enzyme, your reaction starts. So you have to take the reading after 5. So you will be reading, once you add the enzyme, that's a 0 minute. You have to read the absorbance here. Then after 5 minutes, you have to read the absorbance for the same cuvette. Then another 5 minutes, another 5 minutes, another 5 minutes. So you can basically, like you guys are working in a team, you can coordinate how you do that. So basically you can add the enzyme to this and measure its 0 time period, and then you have to measure for this cuvette after 5 minutes. So you can maybe start the second one, and you have to be very careful that you have to keep track of time. So you can add, like when you add the enzyme to first one, that's your 0 minute. Now you have to take the reading after 5 minutes. So you have 5 minutes in between. So you can start the second tube. You can, in these 5 minutes, you can add enzyme to this end result. And then after so these work in a team and work accordingly. But remember that once you add the enzyme, your 0 minute starts, the time is running. So be careful with that. So you will in the lab, you have to just put the data here. And after that, what we have to do, I will explain in the lab. So these are all instructions for the lab. I hope you can understand it, and we will work together in the lab.